Is attached to a G protein-coupled receptor. A G protein within the cell then becomes activated by gaining a phosphate from an ATP and switching from GDP to GTP. The G protein then activates an enzyme, and the G protein loses its phosphate and returns to GDP.
Now transduction occurs through a process called phosphorylation cascade. First, the activated relay protein activates the inactive protein kinase by giving the kinase a phosphate. Then, the now active protein kinase 1 grabs ATP floating in the cytoplasm due to cellular respiration and takes a phosphate from ATP, which then becomes ADP, now with one less phosphate. The active protein kinase 1 transfers the phosphate they took from ATP to another inactive protein kinase and repeats the same cycle. Also, during this process, the protein kinases eventually have to revert back to being inactive when the cellular response has been carried out, and by doing so, a protein phosphatase removes the phosphate from the active protein kinase to reduce the kinase to being inactive. After the last protein kinase, which in this example is the third one, obtains a phosphate from ATP, the protein kinase 3 delivers this phosphate to an inactive protein this time, so therefore there would be no more kinases attaching proteins to other kinases. Once the phosphate is attached to the now active protein, the active protein travels to the ER and binds to a ligand binding site to tell the ER to produce calcium ions, which is the last step, the response.
In the tyrosine kinase receptor system, two signal molecules attach to two receptor tyrosine, which then join together to create a dimer. This requires 6 ATP that will become ADP once they give one phosphate and activate the 6 tyrosine. A relay protein attaches to those tyrosine kinases and is activated by the phosphate, setting the tyrosines back to ADP and inactive.
Now transduction occurs through a process called phosphorylation cascade. First, the activated relay protein activates the inactive protein kinase by giving the kinase a phosphate. Then, the now active protein kinase 1 grabs ATP floating in the cytoplasm due to cellular respiration and takes a phosphate from ATP, which then becomes ADP, now with one less phosphate. The active protein kinase 1 transfers the phosphate they took from ATP to another inactive protein kinase and repeats the same cycle. Also, during this process, the protein kinases eventually have to revert back to being inactive when the cellular response has been carried out, and by doing so, a protein phosphatase removes the phosphate from the active protein kinase to reduce the kinase to being inactive. After the last protein kinase, which in this example is the third one, obtains a phosphate from ATP, the protein kinase 3 delivers this phosphate to an inactive protein this time, so therefore there would be no more kinases attaching proteins to other kinases. Once the phosphate is attached to the now active protein, the active protein travels to the ER and binds to a ligand binding site to tell the ER to produce calcium ions, which is the last step, the response.
Next is the insulin signal transduction pathway. First, the insulin molecules must move into the tyrosine kinase receptor proteins. Once attached, the proteins begin moving together and form a dimer. Next, 6 ATP is converted into 6 ADP so that phosphates can phosphorylate the tyrosines. Once activated, the IRS-PTB molecule binds to the PY972 part of the receptor and becomes phosphorylated. PI3K-SH2 domains bind to the molecule and become phosphorylated. P110 of the PI3K molecule leaves to the plasma membrane. Then, P110 phosphorylates the PIP2 molecule to form PIP3. PIP3 molecules attract AKT and PDK1 to themselves via pH domains. AKT is phosphorylated by PDK1. Then, mTORC2 phosphorylates AKT again. AKT is used to phosphorylate GSK3. GSK3 creates PP1, which activates GS, which then produces glycogen. But what if insulin is unavailable? An SHIP2 molecule is used to dephosphorylate IRS-PTB and the tyrosines. Then, SHIP2 and P10 dephosphorylate PIP3 to form PIP2. No PIP3 means no AKT, which means that GSK3 will not be phosphorylated. GSK3 is unable to produce PP1, which means that glycogen is not made. When